Hello. Hello and welcome to this webcast, Free Energy Perturbation, How Computational Chemistry Saves Time in Drug Discovery, hosted by Scientific Computing World and sponsored by Cresset. My name is Robert Rowe. I'm the editor of Scientific Computing World, and our guest speakers today are Dr. Sofia Bariami, Product Manager of Flare FEP at Cresset, and Dr. Barbara Zalziska, Assistant Professor at the Vrije University, Amsterdam. Sofia studied applied mathematics and physics before completing her PhD in computational chemistry at the Michel Group at the University of Edinburgh. Following the successful completion of her Cresset postdoctoral program, Sofia currently holds the role of Associate Product Manager. Her primary focus is on Flare FEP. Barbara obtained her PhD from the Maastricht University in the Netherlands, where she investigated protein-protein interaction inhibitors. She then completed her postdoc at USC Los Angeles in the lab of Seva Kartvik, focusing on modeling GPCRs. Barbara is now Assistant Professor at the Vrije University, Amsterdam. Free energy perturbation calculations have become a powerful tool to perform accurate prediction of binding free energies. In this webinar, the speakers will explore the benefits of FEP methods in computational chemistry and identify the challenges associated with the application of this method. Dr. Bariami and Dr. Zalziska will cover the benefits of FEP calculations, including the optimization of drug candidates, the method's precision, and potential cost savings it can bring to laboratory procedures, the challenges of running an FEP calculation successfully, the assessment of project data suitability, the effect of correct preparation of the molecular system on the results of the calculations, and how to address the challenge of the demand of computational resources. The speakers will also introduce Flare FEP Cresset solution for accurate relative binding free energy predictions as a way to tackle these problems and highlight the most recent enhancements incorporated into Flare FEP. After the presentation, there will be a quick question and answer session, which anyone can use to ask the speakers a question. If the speakers do not have time to answer your question today, the information will be passed along so our team can follow up at a later date. Sofia, Barbara, over to you. The floor is yours.
Thank you very much for the introduction, Rob. So yes, today we're going to talk about how free energy perturbation calculations can accelerate drug discovery with the prediction of molecular binding affinities. So first of all, I would like to introduce Cresset. So Cresset is a company. We are based in Cambridgeshire, and we're doing software development for drug design. So we are collaborating with a wide range of innovators in academia. And what we do is that we make accessible graphical user interfaces on top of the complex science of our collaborators. Our software, our applications are known for their user-friendly character, and they're very modern, flexible, and our aim is to make them accessible for everyone. From the software that Cresset offers, today we're going to focus more on Flare, which is our ligand-based and structure-based discovery platform, and specifically on Flare FEP, which is our platform for free energy perturbation calculations. But other than Flare, which is our big main software, let's say, we are also offering solutions for biosteric replacement like Spark that gives excellent results. It's proven in publications, and also Blaze, which is our virtual screening engine. If you want to, you can use Blaze to increase your wet screening hit rate very fast. So talking about FEP calculations, so the implementation of FEP in Flare builds on top of the best open source tools. And what we do is we combine them with Cresset expertise to deliver a very easy to use software. We're in collaboration with Professor Julian Michel at the University of Edinburgh. in Julian's group, they are developing SIRE, which is the molecular dynamics engine we are using to run FEP calculations. We are also using the different open source tools, as you can see here. And what we do is that we create a very friendly user interface in Flare in order to be able to use the software. So we've made a lot of progress the past years. The latest release of Flare FEP came with Flare version 8, and now we're actually able to also do charge perturbations. At the same time, Cresset is a member of the OpenFF consortium, so any force field that is developed in this consortium automatically gets into the software after a bit of testing, of course. So now we'd like to pass on to Barbara, who is going to introduce herself as well and talk to us about what she's doing at the Vrije University, Amsterdam.
Thank you, Sofia, so much. My name is Barbara Zaczycka, and I work at University of Amsterdam in the Netherlands, and I lead the computational medicinal chemistry group within the medicinal chemistry division. And in this division, we have two main research lines, one which focuses on G protein-coupled receptors, in particular on the chemokine receptors and histamine receptors. And then second research line that we focus on is that fragment-based drug design. And we combine within the department, we have expertise both in biochemistry, molecular pharmacology, as well as design and synthesis. And of course, the central role is the computational part. So as mentioned before, the computational part plays the central role in our department, working hands in hands with the synthesis, as well as molecular pharmacology, molecular toxicology, and biomolecular analysis. And in the computational medicinal chemistry group, we use both structure and ligand-based techniques, where we can advance heat identification, heat optimization, and as well as lead optimization, leading to preclinical development. And within those three main parts, we now try to also explore the free energy perturbation methods, and that will be one of the case studies that I will present today.
Perfect. And talking about free energy perturbation calculations, I would like to give you a very short introduction in case there are people in the audience that don't know exactly how this method works. So as Barbara said, let's imagine that we are in the heat optimization stage, and we have a very typical drug discovery problem where we have a nice ligand, we're very happy with it, we know its activity, we also know how it interacts with the protein, maybe we have some X-ray structures, right? And we're at the stage that we want to optimize this ligand. We would like to do some modifications, let's say. So for example, as you can see here, what I did is that I changed a little bit the scaffold to include a chlorine. So since these two ligands are quite similar, we assume that ligand B, the new ligand that we have not synthesized yet, has a common binding mode to ligand A. What we were trying to do is predict the activity of ligand B without having to synthesize it first. And this is what free energy perturbation does. It helps us predict binding affinities of ligands that share a similar structure to a ligand of known activity. So just to put it a bit more in context to the FEP terminology, imagine that if you have a way of accurately calculating or predicting the delta G of bind for A and for B, or at least the difference between these two activities, then we could actually calculate the activity of B. So the question we're asking here is, what is the relative binding free energy of B with respect to A? Unfortunately, this direct calculation of delta G are very complex. There are like many different factors that contribute to this, to the protein ligand binding, like the direct interaction of the ligand and the protein, desolvation effects, strain energies associated with adopting conformations associated with binding. And that's why this calculation is computationally too expensive and cannot be done accurately. And this is where alchemical free energy calculations come to the game, because they can help us assess this binding affinity. So in order to run free energy calculations, what we're using is a thermodynamic cycle, as we call it. So this cycle is used to estimate the free energy between difference between two molecular states. Okay. The basic idea is to create a closed loop, as you can see here, or like a cycle, as we call it, which involves a series of transformations between the initial and the final states. By considering the free energy changes along the different paths of the cycle, we can derive the free energy difference between the initial and the final states, even in the cases where direct measurements are very challenging, as I said before. So in essence, instead of calculating the horizontal processes, which are very challenging, what we do is that we calculate the vertical processes. And as you can see, a vertical process involves a transformation both in the free state, so the ligand in water, but also in the bound state, so the ligand bound into protein, which is in water. So transformation from ligand A to ligand B. And now the question is, a transformation sounds good, but how are we going to do it? And the answer is through a switching function lambda, which varies between 0 and 1. So when this function helps us morph this ligand through like a series of intermediate states and go slowly from ligand A to ligand B. So for lambda equals 0, we are in state A, so we are looking at ligand A, and for lambda equals 1, we are looking at ligand B. So the question now is how we are doing this transformation, how we're using this lambda parameter. So we know that in order to describe a molecule, we are using a force field, right, which is a set of parameters and functions to describe the behavior of the molecule. So the lambda parameter actually changes a bit these parameters, the force field parameters. So, and this change is done very gradually. So let's get an example. If we are at lambda equals 0.05, we have 95% of parameters of molecule A and 5% parameters of molecule B. So you can see how this transition happens very gradually. Now, for each lambda parameter, we have a molecular dynamic simulation. So for each lambda parameter, with the specific parameters that are derived with this switching function, we are running an independent molecular dynamic simulation. This means that we have one MD simulation for every single lambda window we have. Now, the more lambda windows you have, the smoother the transition is going to be, because what we're doing is that we're calculating the free energy difference between adjacent windows, and then we're adding up everything in order to calculate the binding free energy from A to B. So you can understand that the more lambda windows we have, the more MD simulations we have, so the more computationally expensive it is. However, you cannot take very big steps, because then the calculation does not converge. Then the results you're getting are not accurate. You may have big error bars, so you're not sure about your results. So you need to find this balance between accuracy and computational cost. Thankfully, in Flare, we have developed an algorithm that optimizes the amount of lambda window you need, so you can have very accurate FEP calculations with the minimum amount of lambda windows possible. Another thing that we need is a connection between the molecules. So we need a way to define these transformations. Because if you have two ligands, if you have various ligands, it's fine. But what happens if you have tens or like hundreds of ligands, right? So another very basic part of an FEP calculation is the FEP graph, as we call it. So it is a graph that includes all of the molecules of our project. You don't need to have just like one transformation. You can have like multiple. And what this graph does is that it finds the best connections between all the molecules of our project. And it also, you have the option to create cycles, as the one that I'm showing here. You have an option to create a star graph, as we call it. So you start from a molecule in the center with a known activity, and then you just do modifications. You connect this molecule with the rest of the data sets just with a single line for transformation. And this is how you get the FEP graph. At the same time, there are cases where a calculation may be a bit difficult to happen. So let's say if I'm changing a large number of atoms at the same time, maybe the results will not be that accurate. So what FLAIR does in FLAIR FEP is that it will generate an intermediate molecule, not to be confused with the alchemical intermediates that we do in the method for FEP calculation. This is a new ligand that is actually predicted and inserted in the graph. And as you can see it here with the gray background. So this is inserted into the graph in order to help the software do the calculation. Instead of going from A to B with the very transformations, you go from A to the intermediate molecule, and from the intermediate molecule to B. At the same time, when this graph is generated, you have some link scores that you can also see here. So it's this number above each link, which gives you the probability that the FEP, the transformation of this specific transformation will work. So the bigger the score, the easier the transformation. So this can help you, first of all, troubleshoot your FEP graph. Maybe you are maybe you have a project with some challenging transformations. If you see that the link score is quite low, then maybe you can insert an intermediate or think about other connections that you can do in order to run this calculation successfully. And the another question is where we run these calculations, right? So as I said, for each lambda window, you run one MD calculation. However, this makes it impossible to run locally because you need hours of calculation. So what you need is usually a cluster of GPUs where the calculations run in parallel at the same time. So you can save a lot of time. In FLAIR, in Cresset, we offer you different licensing options where you can use your own cluster. But even if you don't have your own cluster, then you can use Amazon Web Services instances to run your calculations there. So this is more or less what happens. This is more or less a summary of what FEP is. However, now if we look at a real project, let's have a look at what exactly we need in order to run an FEP calculation. First of all, what you need is a protein crystal structure. So the FEP calculations are structure enabled. You need to have a protein structure. What I'm showing here is a GPCR, so it's a P2Y1. In FEP, in FLAIR FEP, you can run calculations on membrane proteins, and actually recent studies that we've run internally have shown that the results are actually very good. They're better than results that have been reported in the literature so far with other software. So you have your crystal structure, and then the next thing that you need is a series of congeneric ligands. And when I'm talking about congeneric ligands, I'm talking about ligands that share a similar scaffold. but they just have differences in specific R groups. So here, for example, you see that we have differences in the places in R1 and R2 on the right-hand side of the ligand, but also in R3, R4, and R5 positions on the left-hand side of the molecule. Okay. Next, we need to make sure that the quality of our structure is very good. The thing is that the accuracy and the reliability of the result depends significantly on the quality of the initial crystal structure that we used as a starting point for our calculations, right? What is the most important bit is the binding site. It's where the ligand interacts with the protein, so it needs to be very, very accurately represented. So crystal structures with high quality provide a very reliable starting point, ensuring that the ligand is placed in a very biologically relevant position within the binding site. But at the same time, if you have a crystal structure where the quality is not great, what you can do is use the electron density maps to get, information about the distribution of the electrons of the molecules, and maybe make some modifications, maybe make sure that you represent the binding pocket as best as possible. Another very important aspect that we need to pay attention when we are preparing our system before clicking on the button to run the FEP calculation is hydration. We need to make sure that we have all the water molecules that should be in the binding site. Water molecules are very important because they can form interactions between the ligand and the protein. So they work as a bridge, let's say, between these two molecules. We need to make sure that the water molecules that are there are happy, that they are thermodynamically happy, as we call them. And in FLAIR, you have a few methods to assess how good the hydration is, especially around the ligand in the binding site. First of all, we have a method called DC&CMC, or Grand Canonical Non-Equilibrium Candidate Monte Carlo. So what this method does is that it uses Monte Carlo moves to insert and delete waters within a specific sphere, a sphere that you can that is defined with the help of the ligand. It surrounds the ligand, but it can also surround. You can also choose where the center of the sphere can be. So you can adjust the radius, you can adjust the center. And during the calibration, what it does is that it removes and inserts water molecules, and then it accepts the water molecules that satisfy specific criteria. So this ensures, so if you have, let's say, a binding site that is hidden inside the protein, or you think that the crystal structure has not captured well enough the water molecules, then DC&CMC is a very good method to use at the beginning of the calculation before running the production. Another method that we have in FLAIR is the 3Dism, or 3D reference interaction site model, which is a modern approach to solvation. So conceptually, it is equivalent to running an infinite time molecular dynamic simulation on the solvent, keeping the solute fixed, and then extracting the density of the solvent particles. So this gives you the places where a water molecule will be happy to be. And just because it gives you the place of the oxygen, what you can do is create a nice water molecule and use this water molecule for your calculation. And I really like 3Dism because it uses Cresset's proprietary Z force field that models electrostatic interactions very well. So in the GPCR case study that I was talking about, where we have the very good results, this is the method that we used in order to get hydration in the binding pocket. And it actually, I think this is the why we got so good results. Finally, another very important aspect to look for when you're running an FEP experiment is the ligand alignment. So you have, as I said, we have a series of congeneric ligands. What you want is these ligands to be very well aligned, okay? Because, sorry, the alignment and the overlay of these ligands affect how optimally the perturbation network, so the graph, the FEP graph, is going to be set up. So this defines the perturbation between the ligands, so we need the optimal graph there. There are a couple of ways to do this alignment. A lot of people are doing docking, so they prefer to dock all of their ligands in the binding site. However, we have seen that in our case, it's better just to run a conformational hunt and align experiment with a substructure alignment, as we call it. So we have a very tight binding of the different ligands. So my suggestion would be, if you have a crystal, a co-crystal ligand, align all of your molecules to this one, because you know that this is like the best binding pose of the that your molecule can adapt. If you don't have one, you can do a docking experiment. Maybe choose the molecule with the best docking score, the one that you like the most based on your intuition, and then again use conformation hunt and align in order to align all of the rest of the molecule on the specific, on your favorite pose, let's say. But it's very important to have a very tight alignment on this. And these are all advice that you get from benchmark data sets, data sets that have been published a lot and that we're using in order to benchmark our software, make sure that it actually works well. However, in real life, projects can have a lot of challenges, right? It's not so straightforward. So again, I'm going to pass on to Barbara, who is going to talk to us about her work at Vrije University Amsterdam and the challenges around preparing a system for an FEP experiment.
In our case study, we are working on histamine H1 and H4 activity antagonists, and we are interested in polypharmacology. The first target is the histamine H1 receptor, and we have the X-ray structure available. However, it is co-crystallized with the toxic molecule, and our congeneric series of ligand has a different chemotype. Therefore, the challenge in this in modeling this protein was to predict new binding poses for the ligands. In the second protein target, histamine H4 receptor, we encountered even bigger challenge. At the moment, there is no available inactive state structure of histamine H4 receptor. Therefore, we decided to build the homology model and predict the binding poses of the series of our congeneric ligands. And if we look at those series of congeneric ligands, we have 39, both active and inactive for histamine H1 receptor and H4 receptor, with the span of PKI activities with a difference 3 log unit, which accounts for a very good starting point for the FEP. However, during the alignment step, we encountered the challenge as our predicted binding poses showed different poses for different ligands, and we had to encounter for it.
So yes, as Barbara said, the challenges around in a proper real world experiment are a lot, especially when you don't know the binding pose of the ligand that you're working with. But now I would like to talk a bit about other challenges that a researcher may face and how FLAIR FEP can address them. First of all, there is the challenge of usability. Many FEP solutions that are out there in the market are difficult to use. Sometimes you need to, they require scripting skills. You may need to do some like manual work or be very adept to writing bash scripts in order to optimize this process. However, Flare FEP offers a very user-friendly interface, so we try to solve this obstacle by the very user-friendly interface. So as you can see here, what I'm showing you is how the interface looks like. What I have is a few 2D ligands, so they don't have a 3D structure yet. What I do have though is a co-crystal ligand. So what I'm going to do is select all of my ligands and then choose the substructure alignment, as I said before. So what Flare is doing at the moment is that it generates, it aligns all of the 2D ligands on the co-crystal ligand. Then I'm going to click on FEP, create a new FEP project. So here you see the different options you have. I'm selecting to add intermediates in the FEP graph. And if I wait a little bit, you will see that Flare will create the FEP graph. Just like that, where the link scores. Then the next step will be to just click on Run FEP, select the force field or change other parameters, and start the calculation. Another challenge is the post-processing. So all is good. I run the calculation finished. What do I do with this now, right? Sometimes you need to have a deep understanding of statistical mechanics and thermodynamics in order to understand these results. However, in Flare FEP, we have very intuitive analysis and troubleshooting tools to help you analyze your results. So what I'm showing here is how you can access a benchmarking experiment. First of all, you have the 3D window where you can see the ligands at the beginning and at the end of the transformation for forward and backward transformations. Then you have the activity plot. If you have experimental data, you can have this nice scatter plot that shows you statistics. The overlap matrix that has to do with how many lambda windows you're using. Cycle errors to assess whether there is hysteresis and you need to troubleshoot. Link plots about the convergence of the calculations, torsion plots that shows you the different torsions of the molecules and how they behave throughout the calculations, convergence plots for all the different parts of the calculation, contacts, where you can monitor ligand-protein contacts and also report their frequency throughout the whole lambda schedule. And also, finally, you can take some notes if you want to about the specific link that you are working with. Another challenge is the computational resources. So not all researchers or not all institutions may have equal access to resources, to GPU resources. However, with our new flexible licensing options for Flare FEP, we can give you access to Amazon Web Services, which is a very easy way to run your calculations there. You can literally choose the number of GPUs that you would like to run. You can have a small cluster or of around, let's say, up to 30 GPUs, or you can have very big clusters, more than 200 GPUs. So depending on your needs, we can tailor our offering to make sure that we satisfy your needs and make sure that your project runs optimally, actually. Finally, preparing the system for an FEP calculation may not be trivial. Running the calculation itself may not be trivial. Imagine that you can run a benchmark run is when you have some experimental data as well. So what you do is you run FP calculation to get the predicted data, and then you get some statistics to actually define how good your FP calculation is, how good your model is, and what to expect when you actually add some new designs in there. So running an FP calculation, people consider it not to be a very trivial task. However, with Flare FEP, you can have access to our FEP Knowledge Hub. It comes with training, and also a dedicated application scientist is going to help you get started for the first year of your license, going to be there to make sure that you're using the software correctly and have the best results possible. Finally, I would like to show you how Flare FEP has improved throughout the years and throughout the releases. It's one of our products that we really want to make sure that we're adding more and more functionality, and we make it better with every single release. So what you see here with the shades of blue is our performance from Flare v5 2 years ago until Flare v7. As I said, we released Flare v8, but the benchmarks are still running. That's why it's not included in the specific graph. And what you see is the R-squared, so the correlation between the experimental and the predicted activities. Obviously, the closer to the bigger, the better. And at the same time, on the right-hand side, you see the mean unsigned error, which is something we want to drop. And as you can see with the different releases, these two statistics are improving. At the same time, we are comparing here our results with the Wang data set, which is a data set that's very widely used for benchmarks. And we see that we are doing very well, and we keep improving. In the last release, as I said, we are able to do chart transformations, so go from a neutral ligand to a ligand with a chart. You're able to do to use membranes in your system, so use it for GPCRs, and we are, we're very, very optimistic for the future and for what it will bring to Flare FEP. So with this, I would like to thank you. If you are interested in our case studies, if you want to learn more about Flare, about our software, or Flare FEP, please do contact us. You can also subscribe to our newsletter. I promise we won't spam you a lot. Yes, I'm happy to answer any questions that you may have. Thank you very much.
Thank you very much for your informative presentation. We will now move over to our short question and answer session. The first question I have, you talked about benchmarking Flare FEP on GPCR targets. Do you have any results that you can share with us which show the accuracy of your method?
That's actually a very good question, because I'm pretty sure our Jenny Brooks, one of our application scientists, have published some results recently. Give me one sec. I think I can show you some results. Okay, can you see my screen now?
Yes.
Perfect. Yes. So as you said, usually what we do is that in every release of the software, we run the Wang dataset, which is the standard benchmarking dataset. At the same time, we want to do something more challenging, especially after introducing membrane FEP in our calculation. So what we did here is that we ran 3 benchmark runs for these 3 GPCR proteins that you can see. So we're talking about A2A, OX2, and P2Y1. These are all proteins that have been also benchmarked by Ross et al. Using other software that are in the market at the moment. And what you can see here is that we're actually performing very well in terms of correlation. So the R-squared, as you can see in all 3 cases, it's greater than the published results. So we see a correlation of around 0.5, 0.6 in all these cases, which for a membrane protein is very good. especially in the case of P2Y1, we're talking about a binding site which is quite exposed to the lipids as well. So it's a very challenging protein to model. At the same time, you see that the mean unsigned error is quite low. You're expecting the mean unsigned error to be at around 1 kilocal per mole. Same for tau, and same for link-wise RMSD. We're saying that it's smaller in the first two cases, in A2A and OX2, and a bit larger for P2Y1, but this was the challenging one, as I said before. So yeah, we're really happy with these results. I think it's like very exciting, and because GPCRs and in general membrane proteins is a big part of like the proteins that people want to study actually. So we're really happy when we got these benchmarking data sets and results.
Good stuff. And there's a question here about having a good quality crystal structure would be great, but what do you do if you have a model from AlphaFold?
Oh, yes. Well, AI is everywhere, right? So yes, AlphaFold is used a lot in order to predict crystal structures of proteins. The problem with AlphaFold is that, well, first of all, they don't include any ligands bound to the protein. So AlphaFold predicts just the protein structure in the apple, let's say, so without any ligands. So usually, when you have a ligand, a protein in the presence of a ligand is kind of like altered, you have an induced fit phenomenon. So the problem there is that probably the structure that you're going to have may be a good starting point. At the same time, you need to be sure about the interactions that are happening in the binding site. And if there are these conformational changes, then this is something that is only captured through experiments and not through a model generated by AI. At the same time, you need to model the binding site very accurately. You need to be, as certain as possible of what are the residues that are in the binding site. And I'm not sure whether there is any method to test the results of AlphaFold. Probably, if you have a similar protein, what you can do is look at the PDB database and maybe look for similar proteins, see how maybe the binding site or the structure is in general, and compare it. But yeah, I think it's important to see AlphaFold structure as a starting point. Mm-hmm. But then maybe you need a bit of like refinement, let's say, in order to use it for like FEP calculations. But if you have it, you can run an FEP. I mean, it will run, the calculation will run. I'm not sure how accurate the results will be at the end.
Okay. Can you do absolute free energy calculations? Oh,
that's a good question. So what we're talking about was relative free energy calculations, right? A relative is when you transform a ligand A to ligand B. Now, absolute FEP calculations is another thing where you are literally looking at individual ligands and you try to assess the binding affinity of each of these ligands. We have validated the method. Our science team has validated the method, and we are planning to put it into the software next winter, probably. But it's still, it's not in the software right now, but it's in our plans to include it. I think it's going to move one Flare FEP one step further, because you're going to leave, let's say, the heat, the modification of the ligand, let's say, and go to the heat identification, where you'll be able to screen, let's say, different types of ligands, and then assess whether they bind well or if they bind strongly or not. But this is on our to-do list. It's coming in probably in 2 releases from now. So yes, looking forward to it for sure.
Okay. What about evaluations of the software? Do you offer them? And if yes, how does it work?
Yes, we do. So if you're interested in evaluation of the software, then please contact us. You can find my email, or you can email Cresset support. So there are our emails on the website. The way it works is that we give you license for 1 month. We help you set up everything, and then you can use, you can run FEP as a benchmark in one of the projects that you already have. You can use it in a prospective study. It doesn't have to be something published already, so you can use it for the project that you're working on at the moment. So you have access to the license, to the project, to the software for 1 month, and then if you're happy, then you can you will talk with our account managers to proceed with the license.
Okay. What about what simulation timescales do you recommend through every stage of FEP?
Yeah. So usually the default that we are using is 4 nanoseconds per lambda window for each for transformations with no charge perturbation for normal transformations, and 10 nanoseconds for transformations that also include the charge perturbation. So this is the default. Now, if the, let me share my screen to show you something. If you see that the calculation that you ran did not converge, or you had issues, for example, with a specific calculation. Here, what I'm showing you is a type 2, so it's one of the usual benchmarks. As you can see here, we have a calculation that has a bit of hysteresis. This means that probably there is a sampling issue. What you can do is just change the settings of this link, add another instance. This means it will run the same calculation again, and you can change the simulation length to whatever you want to be. Usually when you have hysteresis, it means that you need to run a bit longer, and this is how you do it. So changing parameters in Flare FEP is very intuitive and very easy to do. But what we're suggesting is around 4 to 5 nanoseconds per lambda window for the normal transformations, and then 10 for the charge perturbations.
Okay. There's a couple of questions here kind of relating to 4090 GPUs, I think the RTX 4090, and there's been some recent good benchmarks or recent benchmarks being run on a single 4090 GPU. Have these methods or Flare FEP been benchmarked with the NVIDIA 4090, for example?
At the moment, we haven't run any benchmarks with NVIDIA 4090. I just know that it's the new NVIDIA driver, so it's going to be super fast. But since it's supported by NVIDIA, our calculations are going to run fine with us. With them.
Yes. What methods did you use to build the homology models?
So I think this is a question for Barbara.
Thank you so much. In my case study, we used ICM and MolSoft to build homology model.
Would you like to come back to this? Shall I move on to the next one?
Sorry?
Shall I move on to the next question?
Yeah.
Yeah, okay. What do you think are the causes for the large error bars?
Right. So large error bars may mean that you have inefficient sampling. sometimes, especially when the ligand is quite flexible, you see that you need to run longer calculations in order to find all the relevant, in order to sample well how the ligand behaves in the binding pocket. At the same time, the error bars that we show in our calculations are not just the error bars of M-bar, which is the method that we're using to derive the final delta G. These error bars are bigger just because they incorporate as well the effect of hysteresis, the propagation of errors. It's the worst case error bars, let's say. Okay. So we're using a method from Zoo to calculate the binding affinities, and the calculation of the binding affinity of a specific ligand is affected by how the map looks like. So let's say if a ligand is connected to other ligands through links that did not work very well, then this will also affect the error bars because of the propagation of errors, as I said before, going from one ligand to the other in order to calculate the difference in the free energy of binding.
Great. Are there any special considerations for binding sites that contain metals?
Oh, metals are a bit tricky, right? So the metals usually require very specific force fields just because of this unique electronic structure and like coordination geometries they have. So in FEP, in FLARE, we're using the amber force field and the open FF force field. So if these parameters are not supported by these two force fields, unfortunately, you will not be able to run the calculations. At the same time, you need to be sure about the oxidation states. You know, some metal ions can have oxidation states that change depending on the ligand that is close to it. You need to think about solvation effects because some metals can be influenced by the solvent molecule in the binding site. Maybe some water molecules can participate in coordination with a metal ion or form hydrogen bonds. So yeah, it's a tricky one. Okay. But yeah, if you're running an FEP with a metal, good luck with it.
Okay. And as you kind of mentioned force fields, so we've got another one. Is FEP based on force fields?
Yeah, so both the protein and the ligands both need to be parameterized by using a force field. For the protein, we're using the amber 13SB. Whereas for the ligands, you have different options. You can either use GAF2, which is the corresponding amber force field for small molecules, or you can use the open force field, the OpenFF. We support all the types of all the versions of OpenFF that are out there. At the same time, if you choose to use OpenFF, then we also support generation of custom parameters for the torsions. So modeling the torsions of a molecule is very, very important. And sometimes the parameters that you get from transferable force fields are not great, unfortunately. So if you have a molecule that you know that the force fields that are out there will not describe it sufficiently, for example, the azethidine residues, we know it's not very well described, neither with the amber force field, neither with the open force field, then you can derive custom parameters for the specific one and run your calculation using these new parameters.
Okay, I've got a question here. In the calculations, you take into account the presence of the water molecule interaction between the target and the protein, or do you take into account the presence of the water molecule?
So both. Okay. the FP calculations run in, let's say, 2 different legs. So the first leg, you only have the small molecule solvated in water. So there are obviously interactions between the small molecule and the water. The second leg is the small molecule bound to the protein, which is also in a water box. Solvation of the binding site is very important, as we said, because yes, there will be water interacting with the ligand. Sometimes these interactions are very important because they work as a bridge between the molecule, the small molecule, and the protein. So we definitely take these into consideration, and this is why it's very, very important to do a very good preparation of the system. Make sure the solvation is very good, and this is why you can also use the GC and CMC method that deletes and insert water molecules around the sphere to make sure that the molecule is very, very well hydrated.
Okay, and does FLAIR use an enhanced sampling method like REST2 for better convergence?
It doesn't use an enhanced sampling method at the moment, but it's something that we're looking to include into the software in the future. As you probably noticed, FLAIR FEP is quite a new product. So with each release, we're trying to include more and more features in order to give better, more accurate results. But enhanced sampling is not in the software right now, but it's on our to-do list for sure.
Okay, great. So something to look forward to in the future.
Exactly.
Okay, that brings us to the end of today's webcast. The content will remain live on the Scientific Computing World website at www.scientific-computing.com for the next 12 months. So please spread the word if you have enjoyed the presentation. Of course, please note the live question and answer session will not function if you are listening on demand. Once again, thanks to our speakers, Sophia and Barbara, and of course to our sponsor, Cresset. Thank you all for listening, and goodbye.
Thank you very much. Bye.
Bye.